Well, good evening, everyone. Bottom line is, why are you all joining us tonight? You're here for one reason or another in an association with CMT. You either have a loved one that has it, you have a family member that has one, CMT, or you yourself have it. That's why I'm here. I do have CMT, and I was the recipient at the age of 16. My main reason is, and from my main message tonight is, I want to let you all know that I'm grateful for your support for this organization. You have choices in your philanthropic endeavors, and you couldn't have picked a better investment in donating to the CMT Research Foundation. I'm saying that because in a short 2 years, what Susan and her team has done, we're going to give you some evidence that's overwhelming. A few great key points tonight. First, the investment you made or you helped us make in DTX. DTX is a small biotech firm in San Diego that Susan pretty much single-handedly talked into focusing their technology on CMT. We made a $125,000 investment, and in very, very short time, 800,000 more has been put in by the company. And as of now, and you'll hear this later, they're doing a multi-million dollar raise, and I'm talking some serious money, 50 to 100 million, with CMT therapy is a priority product. Next, we have three projects that we are currently funding. And in 2021, next year, the second round of the funding from the foundation is up for debate. What does that mean? It means that all three projects, the initial results were positive. So the goals of the projects were met, the metric, the milestone, and they're testing, and what they're testing is working for CMT. So that's what it's about. The way we look at the world of 501c3 and philanthropic giving on a disease-based organization is, look, we put our money where our mouth is. We start the ball game rolling. A foundation like us demands a business approach with accountability and a feedback loop. So we maximize the capital that's going into these projects. And we make those investments with your hard donations to the tune of 100,000 to $1 million. And after a success metric is met or a milestone is met, the next part of it goes forward, where pharma invests their own money alongside us. And then obviously we want to move as quickly as possible with the success in the clinical trials before FDA approval. I got to tell you, these small investments we make in these biotechs are really the kindling wood, if you will, in a whole chain of events. So CMT is a rare disease that's grossly underfunded and starved for cash, and starved for people willing to jump in at the beginning. Your donations and your help is changing that dramatically. Finally, I want to tell you that we've got 2 patents in a short time into application process. So we took an idea to a patent application in less than 18 months, and you'll hear from our experts here, that's pretty unheard of. If you don't know it already, it's lightning fast. It points to the quality of our projects we fund, and quite frankly, being shaped by our world-class scientific advisory board. You'll hear from 2 of them today. I couldn't be more thrilled with the progress. This was a white paper idea about 2 years ago that Susan and I hatched up. Our passion for finding a first treatment is unmatched, only by you all who are helping us. I'm actually thrilled with where we are in just a short 24 months. And now it's really the time to ratchet that up. We've got our goals set. This organization is singly focused on delivering a treatment and cure to the CMT community. And that's why you all are here tonight, which I'm really grateful for. And we're getting close. I got to tell you, a lot of people who know me know that I'm a firm believer in success metrics. There's a couple other diseases out there that have a lot of similarities to CMT. Cystic fibrosis being one of them, a great success story in where they funded projects much like we are, demanded accountability, had an economic component on the successes of those projects attached to the foundation. And lo and behold, 3 years ago, they sold their portfolio for $3.5 billion. Most importantly, they've extended the length of longevity for a cystic fibrosis patient, doubling it from 20 to 40 years. And some of the treatments that are going on now are actually giving everyone a normal lifespan, which is incredible. Another one is spinal muscular atrophy. You're going to hear a little bit about that tonight. And it's close, and it's a genetic disease. They have three therapies. Just a few years ago, they had none. MS has 17 different therapies, MS being the CMT of the central nervous system. ALS has some therapies, and there's no reason that CMT can't be next on the list. I know in my heart of hearts it can be done. It just needs the resources to get there. Right now, we're on a trail to raise between now and year end about 1.2 million as an immediate objective. This is going to ensure our re-up for the projects that I had mentioned and you'll hear about. This will allow us to even go into other projects that we're seeing that are coming from some of the big boys out there. All of this leads to the approved therapies that is our end goal. You know, I'm asked, how long does it take to get a therapy out there? And COVID aside, we know with CMT, the science is there. So we know what is causing most of the CMT out there, whether it's 1A, 2A, the 4s, the X's, etc. It takes dollars. Money and the velocity of that money gets us there, and it gets us there a lot quicker. It's really that simple. And from my standpoint and the people who know me on this phone, you know, I'm not stopping until we find a treatment. We have the right team behind us, both scientifically and from the organization itself. And if we had 10 times our budget right now, we'd be 10 times faster and closer to that elusive treatment, let's put it that way.
I'm a parent of a 25-year-old CMT 1A patient and a board member of the Research Foundation. My husband, George, and I have been with Susan and Pat since 2016. We are both retired from the media business and absolutely 3,000% committed to the success that Pat just described. So that's why I'm here. Pat, I think you made some really excellent points about all that has been accomplished by the folks at the foundation in such a short amount of time. We really can make this happen. And I'm glad to see that the group of believers has grown in the last 2 years significantly. So I just want to make a couple of points quickly. Pat mentioned SMA, spinal muscular atrophy, and he also talked about MS, ALS, and cystic fibrosis. And I want to briefly underscore the importance of their successes for all of us at the foundation. These organizations are examples of a drug discovery process, a model that works, that we know works. We've watched it work in recent years. And that is the model that the CMTRF is built upon. Tired of waiting for the NIH or Big Pharma to focus on finding treatments for rare diseases, patient groups have committed to finding cures. So what happens is those patient groups raise the money to fund the most brilliant scientists and researchers so that they can accelerate breakthroughs and solutions for their diseases. They then engage the biotech and pharma in the drug discovery process and stick with it all the way through clinical trials, FDA approval, and then financially participate in the commercialization of those treatments and therapies in order to be able to fund additional research. This is a model that we believe in, and as I said, and Pat talked about, we have seen it be successful, and that's what we're going to do. So my job is really to tell you about what we're going to do this evening, and also to promise you that we will just take an hour of your time, because we know you have some other things to do this evening. We're going to hear next from our Chief Scientific Officer, Keith Fargo, and then Susan Rudiger will do a little roundtable or Oprah-like interview with two very special researchers who are experts in SMA. Chris and Charlotte, helped bring those SMA drugs through to the FDA approval. So they are experts in many ways and in successfully finding these drugs and treatments. Charlotte and Chris are also both helping us to do that with CMT. So I'm very excited to hear from them tonight. Now, as we go along, I hope that you're all very, very familiar by now with Zoom calls, because we live on Zoom calls, right? I hope you will please type in any questions that you have for Pat, for the foundation leadership, or for the researchers into the Zoom chat feature on the bottom of your screen. We're going to facilitate the Q&A throughout our time together. We want to make sure that we answer any questions that you have. And if we hit our 1 hour mark, we will surely follow up with you afterwards to answer any questions that we didn't get to. I'd like to make a few introductions so that you know who's who on this call with you, and then we'll jump right in. So I've told you who I am, but I'd like to start with the next member of our team, Susan Canavari, our newest board member. Susan, if you'd each introduce yourself, we'll run around the room.
Hi there, good evening. I am Susan Canavari, and like Clary said, I am new to the CMT RF board, and I'm excited to be here. In my work life, I'm an SVP of marketing at TIAA and have spent my career in marketing and advertising. So thank you, Clary, and thank you, team.
Susan Rudiger, our fearless CEO.
Hi, I'm Susan Rudiger, and I am the co-founder of the foundation with Pat. I'm also the CEO of the organization, and like Pat, I have CMT. I actually come from a family with 6 generations of CMT, and in my opinion, that's way too many. So we're here to stop it and not see another generation.
Thank you. And now Keith Fargo.
Hi, everyone. I'm Keith Fargo. I'm the Chief Scientific Officer at the CMT Research Foundation.
Eric, over to you. I bet Eric's on mute.
Eric Fasnacht, I'm the Chief Operating Officer for the foundation.
And Chris Larson. Thanks, Eric.
I'm Chris Larson. I am a professor at the University of Missouri and the co-founder of Shift Pharmaceuticals, a small biotech that focuses on SMA and now CMT as well.
Great, thank you, Chris. Thanks for being here tonight. And Charlotte Sumner, we know you all have very, very long day jobs, so we appreciate your dedicating the time to be with us.
Good evening, everybody. It's so nice to be with you. My name is Charlotte Sumner. I'm a professor of neurology and neuroscience at Johns Hopkins. I take care of individuals with CMT and SMA and do research in both areas. But most importantly, I'm fortunate to be a member of the Scientific Advisory Board for the CMT RF.
Thank you, Charlotte. So now before I turn things over to Keith, please remember, put any of your questions into the chat feature on Zoom. Keith is going to present an overview of the portfolio of projects that we are currently funding. And I just want to say one last time, all of the projects that we invest in are fully vetted by some of the world's leading researchers in CMT who make up our scientific advisory board. And we're lucky enough to have 2 of those with us tonight. So over to you, Keith.
Thanks, Clary. Appreciate that. And good evening, everybody. And thank you for being here with us tonight. there's a lot of exciting work happening in CMT, and a lot of that we're involved with, and proud to be really leading the way in terms of the research that's happening in CMT. As our tagline says, we have one single focus, and that is delivering treatments for CMT, and we're well on our way to doing that. So a lot of exciting stuff that we can share with you. Now, the real treat tonight, though, of course, is to hear from Charlotte Sumner and Chris Larson, with, of course, Charlotte being a member of our scientific advisory board, but both really excellent researchers. And Susan's going to have a conversation with them this evening. So I won't take too much of your time, but I do want to go through a little bit of our research portfolio here. I just have this single slide. Now, we get a lot of projects that come our way, and a lot of them are great projects from researchers who are seeking funding to make sure that they can do this research to get us closer to those treatments for CMT. And it really is our scientific advisory board that helps us vet those projects to make sure that we're spending donor dollars wisely. We never forget that the money that comes to the Research Foundation is donor money, and that's being donated to help us with that mission to get us closer to those cures and treatments. So we're 100% laser focused on that, and do want to make sure that we're doing that wisely. So I'm just going to give you a very high level overview of our current research portfolio, and we won't go into any depth in any of these, but if you do have questions about any of them, feel free to pop that into the chat. And if we don't have time tonight, or you just want some additional time to understand some of our research projects or understand our approach, we'd be happy to set that up. So you can just reach out to Susan and Eric, and we'll set up a meeting with you where I can go into greater depth with any of these projects that you'd like to learn about. What I'm going to do is break these down just into three buckets or categories, if you will, of the kinds of research that we fund. And each one of these is then indicated by a particular icon. And so the first one that I'll talk about are gene therapies. And that's indicated here by a blue double helix strand of DNA. And most of you probably know already that CMT, at the end of the day, is a genetic disease. Everyone who has CMT has some kind of a genetic change. You'll sometimes hear that called a mutation, but some kind of a genetic change that is causing them to develop Charcot-Marie-Tooth disease. And so it's no surprise, really, that a lot of the work that's going into finding treatments and cures for CMT are focused on gene therapies. Now, it wasn't that long ago that gene therapy was almost science fiction. It was the kind of thing that we talked about might happen in the future, but we weren't really seeing in our lifetimes. But that changed in 2017. In 2017, we saw the FDA approve the first gene therapy for marketing in the United States. And so we're now 3 years into this brave new world of genetic therapy. And when we think about genetic therapies, you might be tempted to think about things like correcting genes that have changed. And that certainly is one approach to gene therapy, but you can also increase or decrease the activity of genes, or you can block the activity of genes altogether. And sometimes you're focused on the gene that is faulty, but you can easily imagine other cases in which you might want to increase or decrease the activity of another gene that might also impact on the course or progression of the disease. So I'm going to give you just one example here, and that is down here in the bottom right. You can see we're funding a gene therapy project at the University of Missouri. And it's actually Dr. Chris Larson who's with us tonight, who is the PI on that project, along with another researcher at the University of Missouri, his co-PI. His name is Michael Garcia. And again, we don't have time to get into all the details of each one of these projects, but they're doing something really, really clever in this project called knock down and replace. So people with CMT2E, which is what this project is focused on, have a faulty copy of a gene called neurofilament light. But it turns out that your body needs not just to have the faulty gene dealt with, but you also need to be producing normal neurofilament light to have normal function in your neurons. And so this approach that they've developed is called knock down and replace, whereby they reduce the activity of the faulty neurofilament light gene, but you can't stop there because you need regular neurofilament light as well. So they knock down the activity of the faulty neurofilament light gene and then replace that with another genetic signal at the same time that will tell the body to produce normal neurofilament light so that you can have that normal protein taking the place of the abnormal protein. Now, the second category I'll talk about very briefly these are called small molecules, and that's represented here by a green molecular structure icon. Now, when you hear the term small molecule, that can be a little bit confusing because you might think, well, aren't all molecules small? Well, they are. All molecules are small, but we need to differentiate from the genetic therapies and other biologic therapies as well. And so we can refer to these as small molecules. And if you think about the medications that might be in your medicine cabinet right now, things like Tylenol, things like aspirin, things like allergy pills, all of those traditional medications can be considered small molecules. And small molecules can cure diseases, but most frequently, what we're talking about with small molecules is ameliorating a disease. So potentially slowing or stopping the progression of a disease or stopping the symptoms that a person experiences who has that disease. Just one quick example of that, you can see in the top left of your screen here, we're working with a company called Curistem, who is doing what's called a drug screen in CMT 2A. And in a drug screen, what you do is you test thousands of different drugs or potential drugs on a platform that can tell you whether or not they may be beneficial for a disease. And so that's what we have ongoing right now with the Curistem. And one of the important things I'd like to point out about that project and about drug screening in general is that you can screen not just things that are not yet medications, but you can also screen currently FDA approved medications. And that's one of the things that we're doing with the Curistem. Now that's important because if we find out that one of those medications is beneficial in CMT, if it's already FDA approved for some other condition, then that has a much easier pathway to approval than if you're starting from scratch. And then the third category I want to draw your attention to is building better models. Now, most of you probably already know that before we test medications in people, we have to test them extensively in models of disease. And when we're talking about models of disease, we mean things like cells in a petri dish, or we mean animal models such as mice or fruit flies, or even zebrafish. All of these are models of disease. Now, the models are really only as good as the extent to which they mirror the disease that you see in human beings and the extent to which the models react to treatments the same way that human beings would react to treatments. Now, that kind of goes without saying, but I wanted to set up the fact that we're working with Jackson Laboratories that you can see here on the bottom left side of your screen, about one project in. And we're working with Jackson Laboratories to build what we call humanized mouse models. And in particular with Jackson Laboratories right now, we're working on creating some humanized mouse models of CMT1B. Now, what does humanized mean? Well, humanized simply means that the genes of the mice have been edited in such a way that they produce a human gene. Not just the mouse gene that we're interested in, but the human version of that gene, which that sounds a little science fiction, but it does happen. It is possible. It's done all the time. Actually, two fantastic women researchers were just awarded the Nobel Prize about 2 weeks ago for developing the technology that is used for this kind of work. It's called CRISPR-Cas9. And the people at Jackson Laboratory are using that CRISPR-Cas9 to make these models for us. Now, the important thing really isn't the technology that they're using to make the models, although it's nice to see that won the Nobel Prize. The important thing is that these humanized models should react to potential medications much closer to the way humans do. And that can actually then reduce the amount of time that it takes to get from animal work into human clinical trials. And as Pat and others have mentioned tonight, We are all about speed. It's, the one focus that we have allows us to move quickly, allows us to move fast, executing against that single focus that we have, which is delivering therapies. Now, I've probably spoken enough. As I said before, feel free to reach out if you have any questions, and we can take a deep dive on any of these things that you would like to. But I don't want to keep you any longer from the real treat tonight, which is the discussion between Susan, Charlotte, and Chris. So with that, I'm going to pass it over to Susan. Thank you.
Thank you, Keith. And for anyone who has any questions about those projects, feel free to reach out to me or to Keith anytime. We'll be happy to do a deep dive with you into them. And at the bottom of the slide, you'll see that we actually have 3 more in the queue right now that we hope to get announced fairly soon. So here comes the fun part. And if I was compared to Oprah, that's a huge compliment. I actually do have notes that you're going to see me refer to just to make sure that we stay on time and on target. I am really excited to talk with Charlotte and with Chris today and really do a deep dive comparison into the successes that they've had in the spinal muscular atrophy world and how they're relevant to CMT. I think when we're talking about drug development, we want to take a look at people who've had successes. And so we'll talk about why we're comparing CMT to SMA, and we'll talk a little bit about the road ahead for CMT. But let me do a little bit of an introduction to you. So Dr. Charlotte Sumner, I'm so glad to have her on our scientific advisory board. She is really a leader in the CMT space and in the SMA space. She was involved in the preclinical work that led to the clinical trials for the SMA patients, and she was an advisor to many of the companies running the trials. So she's really well versed in what it takes to get or she's been a witness to what it takes to get drugs to market and seen the clinical application of that. So Dr. Chris Larson, you heard that he is the co-founder of Shift Pharmaceuticals, and we're also funding him at University of Missouri for a gene therapy project. You know, he was the scientific director of Fight SMA, and he was instrumental in getting the SMA drugs delivered as well. And as you heard him say in his introduction, he's now turning a focus to CMT, adding that to his portfolio. He is a member of the scientific advisory board of the Muscular Dystrophy Association, and both Charlotte and Chris are top reviewers at the NIH as well. So we really are talking about industry leaders here. So with 20 plus years of experience in the drug development world for these neuromuscular diseases, let's talk with Charlotte and Chris a little bit about comparing SMA and CMT. But I want to start with that drug development journey. So we know that the journey to drug development is long. We've heard upwards of 20 years, and none of us are excited by that number. So I wonder, Chris, if you could tell us a little bit about the steps to drug development, just at a 30,000-foot view, and what we're facing in the CMT world.
Well, thanks for the introduction. And I think when we're talking about drug development, it is a long process. But what's pretty remarkable is that in the last 10 years or so, that has really compressed from what you hear in the news, you know, this 20, 25-year cycle, to something that when it's really focused, it can get done in a relatively short time frame. But there are things that really help get the ball rolling. What are those? Well, it really helps to have a single gene or to identify the genes that are involved. In the case with CMT, it's a monogenic disease, so it's caused by, or for the forms of CMT, it's caused by one disease-causing gene. So right off the bat, you've found a target. And the reality is that that only gets you so far. After that, you have to make a model, because what you have to do is think about target engagement. You have to be able to test your therapeutics, and you have to move those from a molecular level, kind of the test tube level, into an actual animal model. So along those lines, you're looking for those early signs of efficacy, so target engagement. Does my drug bind where it's supposed to? Does it turn on the gene or turn off the gene it's supposed to? And then the kicker is safety. And I think one of the things that we've learned in SMA is that when you're doing this kind of drug development, it's not simply when you move into humans, you're not recapitulating or you're not just confirming the results that you've seen in animals. Humans are a completely different kettle of fish. And so these are brand new experiments and that they are actual novel experiments, not confirming the animal models. And so it does take a little bit of time, and it takes a little bit of time to kind of adjust your mentality as well. So from a patient's perspective, from a patient advocacy perspective, even though the time is shorter, it's never as fast as it should be. So there's always this sense of urgency when you're working on a disease project. But what really speeds this up, I think, is having the underpinning biology behind the disease causing gene, because it really lays the foundation for what those types of molecules, what types of drugs come out of this. And so when you see an enormous groundswell of basic or kind of foundational biology, it really is the basis for the next step. And in CMT, I think that's really where we are, to move into the translational studies, those preclinical studies that are going to be critical for moving into the clinic. So there has been an enormous amount of background work. And I think that this is really going to be the catalyst for how CMT goes from rare disease that is underfunded, as Pat mentioned earlier. I don't think it's underfunded because it's not ready. CMT is ready, period. It needs the catalyst, it needs the funding. That's going to take it to the next level. And I think the remarkable work from the biological perspective is there. It's translating what's next that is kind of that next hurdle. And I believe that CMT is really ready for that next adventure.
You know, as a patient, that is so exciting to hear you, as someone who's been through this journey, say that we're ready and we're there. It feels hopeful, inspirational. It feels good. And I think that in many ways we're riding on the coattails of SMA. So I wonder, Charlotte, if you want to explain a little bit to our audience here what you saw in SMA, what kind of successes you saw, maybe even start with why we're comparing SMA and CMT, and then maybe where CMT falls in that journey as compared to SMA.
Yeah, well, I mean, I think Chris and I were both really fortunate to be involved in the SMA field, which has seen this success. You know, I take care of individuals who have SMA and also individuals who have CMT. And to me, as a neurologist, I see a lot of similarities. I actually almost think of them as a spectrum of the same disease. You know, they're both disorders that affect nerves. You know, the clinical manifestations, one, of course, is weakness of muscle that arises from a problem in the nerves. And fundamentally, that's shared by both SMA and CMT. They're both progressive disorders. And up until recently, you know, really lacked treatment and lacked, frankly, appropriate attention. And SMA was in this situation as well. You know, when I started in the field in about 2002, I mean, there were about 12 people around the world who were working on SMA. It was tiny. And it just shows you how fast and how quickly we have come in that field. So in terms of where we are, you know, SMA is, as Chris sort of alluded to, SMA is caused by a mutation of this one single gene. And so in the field, we focused on that one gene. And what was fortunate, in addition to the genetic understanding of the disease, is that in parallel, there was enormous progress happening in the ability to therapeutically target genes. So, you know, there has been enormous progress in traditional gene therapies or gene replacements, in what are called antisense oligonucleotides, and also in small molecules. And we were able to take those forward in SMA into clinical trials, and they showed efficacy. So that in the last 5 years, we've actually had 3 different medications that have been approved for SMA, the last just in August. This is an example of a sister pair taken care of by my colleague, Tom Crawford, a sibling pair. Unfortunately, the sister was born prior to treatment being available, and she died at 15 months of life. But her brother, when he was born, we actually did have therapy available, and he was treated at 3 weeks of life. And you can see that he's able to sit here at 10 months on his rocker and the final picture is able to walk. And I mean, this is just a remarkable success. I really believe, as Chris does, that CMT is perfectly poised for a similar kind of breakthrough. And the reasons are that we do understand the genetic basis of the different forms of CMT. As I think probably most of you on the call are aware, there's just been enormous progress in understanding the genetic basis of different forms of CMT. But importantly, in addition to that, there's been a big explosion in understanding in how those genes cause disease, and even validating that targeting specific genes could be therapeutic. So I do believe that we can advance from the preclinical side of the schematic on the left and get us into the clinic. What's holding us back? I think a big challenge, and the CMT-RF is really trying to address this challenge, is really trying to get these really revolutionary new kinds of drugs into the appropriate cells for the peripheral nervous system. And so many of the projects funded by the CMT-RF are funded to develop either better kinds of these sorts of gene targeting drug or better ways of actually getting them in to peripheral nerves. And I think if we can get over those hurdles, we're really we're on our way.
Thank you. I think that really speaks to the power of having a well-heeled scientific advisory board. When we first met as a group a year and a half ago, I challenged the scientific advisory board with what are the biggest barriers to drug development for CMT, and getting into the peripheral nervous system was one of them. And as Pat mentioned at the beginning, you know, we single-handedly focused on companies that were doing that in other diseases, delivering gene therapies, focusing on these delivery mechanisms. And the way I like to describe it is, you know, you've got to give your dog the antibiotic when she's got an infection, but she's not going to take the pill herself. You've got to wrap it in bacon or ham or cheese or something to make it yummy to the dog so she'll take it. That's really what we're doing, is what can we wrap these gene therapies in that the cells, the nerve cells, will want to take. And you know, thank you very much. You and the SAB gave us that charge, and we went out and we're now funding 3 projects focused on delivery and working on one more that we hope to be able to announce here shortly. So that was a key priority. And we're really pleased with the progress, certainly, that DTX has made in that realm. So thank you for that. So I think it's really exciting about the progress that SMA has made. Charlotte, will you speak for a moment about the 3 different drugs? I mean, very high level, and then maybe efforts in the CMT space that are comparative?
Yeah, I think that Keith introduced some of them. I mean, in SMA, again, very high level, you know, we're targeting one gene in this disorder, but it's now been therapeutically targeted using 3 different kinds of drugs. One is a gene replacement kind of drug, a viral gene therapy, where the gene is replaced using a virus. It's sort of like a Trojan horse kind of strategy. The second drug that's been approved is what we call an antisense oligonucleotide. This is an artificial, chemically modified single strand of DNA that's engineered to target a particular RNA in the body. And then the final drug that was most recently approved is this small molecule variety that Keith mentioned, a chemical compound that patients can take by mouth. But remarkably, that chemical compound is also able to target a gene. So, you know, all 3 of these types of strategies are very realistic strategies for different forms of CMT. So what the CMT-RF is doing is investing in each of these strategies. It's investing in moving the ball forward for each of these, because more shots on goal is going to be more effective than just going with one strategy alone. And so, for example, there are projects ongoing to either develop better antisense oligonucleotides or antisense oligonucleotides that have better ability to get into the cells we need them to get into. So that is ongoing. And then in terms of gene replacement or gene therapy, the CMTRF is funding projects to take gene therapy forward in CMT. And then also, finally, to identify novel small molecules that could be effective in CMT. So I think using the SMA example, the CMTRF is really using each of these different therapeutic platforms and trying to move each of them forward in CMT.
Yeah, and we have precedent for approval in the rare neuromuscular diseases. I mean, clearly FDA is amenable to these approaches. And so they were for SMA, so why wouldn't they be for CMT if we have the same kind of data?
That's right. That's right. I mean, that's why SMA was such a breakthrough, that each of these strategies proved to be safe and efficacious in patients with SMA. And there's no reason to think that they won't also work in other diseases.
It's exciting. Thank you. So Chris, I know you're very involved in the SMA patient world. What do you think changed in the ecosystem in SMA? You know, you said in 2002, Charlotte, that there were 12 researchers in SMA, and 18 years later, they've got 3 treatments available. What changed in SMA, and how can we, as a patient group, and we as another similar rare disease, how can we follow in some of those footsteps of success?
Well, the first SMA meeting that I went to was in Philadelphia, and we sat around a singular round table with about 8 people. So that was in '98. So I predate Charlotte a little bit. We had expanded by 50%. So, if we want to go old school. But what you see is that there are so many rare diseases, and they are all incredibly needy of funding. And the reality is that patient advocacy groups really are going to be the driving force here. And SMA in the early '90s, patients were, or parents were basically told to take their kids home and enjoy the time that they have with them. That's not really a strategy. What you need to do is to develop a strategy that is based upon science. You have the basic biology where you understand things, and then you leverage that to move into drug development and preclinical things. And so what you see is that the patient advocacy groups, there are several in the SMA space, and they really all range in what their expertises have developed into, whether it's government relations, kind of the umbrella organization, or the mom and pop car wash. These are all things that really come together to build this coalition. And I think that having advocacy groups that worked really well together, that understood the importance of what they were doing within the broader picture, was really critical. But patient advocacy groups are not the federal government. So you have to be really selective in what you fund. And the way that we used to think about it at Fight SMA was that there's some broken field tackling. So you have to do what you have to do to get things done. And whether that's cold calling a company that develops ASOs that have a unique targeting system for cells that are tough to crack into, that's a perfect example. But it's all about doing what you have to do to address that need, to fill in the gap. And if you can, I think that using the CF model is really good too, in the sense that there are all these grassroots activities, and you build this portfolio, and you can really leverage that into a broader community. And you can see how these become actual drugs that the community rallies behind. And I think that's really important. You have to pick the projects that are really meaningful for the community. And as you do that, you really gain momentum. And so I think that when you're looking at how SMA transitioned from very rare, and I don't mean this in a bad way, but depressing kind of context, it was all about the patient advocacy groups making the right moves and working with NIH, working with the FDA, engaging them. It's not a you do it better than we do it. It's that there's this, there's a complement where all of the groups need to come together, because the reality is rare diseases need help. And so when you get together, you've got NIH recognizing that CMT is important, and that's going to be done by funding good projects that push things forward by the patient advocacy groups. So I think that while there are all these great instances of being able to follow SMA from a scientific perspective, from a clinical perspective, I think you can step back and also use the roadmap of how they interfaced and interacted with the federal government and really move things forward in a way that you would never see before. Because for a rare disease, and there are 568 rare neurological diseases that National Institute of Neurological Disorders and Strokes covers, SMA was picked in 2008 to be the one that was closest to the finish line. Why? Not because they liked SMA, it's because patient advocacy groups stood up and said, hey, we're next. Mm-hmm. And I kind of smell that here. So I think the opportunity exists.
Well, those of you who know me and who know Pat know that we're going to stand up and be the loudest voices, jumping and screaming for attention on CMT. And thank you for mentioning both the NIH and the FDA. For the people attending tonight, if you haven't seen, I do an interview with the director of the National Center for Advancing Translational Sciences at NIH and with FDA, and you can see the kind of conversations and relationships that we at the Research Foundation have with them. The other piece that I'm really excited that you talked about, Chris, was the nimble research funding, but then the investment that we make having a return that we can then reinvest and reinvest and reinvest, the way that Cystic Fibrosis Foundation did in CF. And they announced last year that they had now treatments for every person with CF, and they were doubling down $500 million to deliver cures for everyone with CF. And that's when I knew this was a long journey we had ahead, but that it was doable. So in our final seconds of the interview, I'm going to give each of you an opportunity to answer this question. What excites you the most about where CMT is today? We do only have a couple final seconds, but I'd love to hear your answer, and I'd love for the community to hear what you're most excited about.
So I guess I'll start. I am, again, taking care of folks in my clinic and having seen what's happened in SMA, I want to be able to walk in to my next clinic visit and write a script for the next medication. I think it's coming. But I do want to emphasize what a point Chris was making, which is that most funding streams, there's not enough funding in general for CMT, but most that come via, for example, NIH, the way that money comes is that they wait to receive a grant on a topic and decide whether or not to fund it. So it's a passive process. They can't dictate what critical knowledge gaps may exist and funnel funding to that particular issue. And this is why a committed nonprofit foundation like the CMTRF is so important in really taking things forward, because there is this laser focus on what are the critical gaps that are limiting progress in terms of therapeutic development. And I think that is so important, and we saw that in SMA. And I think with that kind of focus and framing of the problem and investing in those things that are currently impeding that progress, we will break through. And I will be writing that script in my clinic very soon.
Thank you. Chris, what about you? What excites you the most?
I think when you look back, the Muscular Dystrophy Association has been great for a number of diseases, but they've really had a lot of pushback because they've been funding basic research for ages and ages and ages without any therapeutics. And I bring up MDA because over the last 5 or 6 years, there have been dozens of gene-targeted discoveries and clinical trials and drugs that are approved in this space. So you could see this kind of long game that was being played. And it's long because of the process, but also in terms of science and the breakthroughs in the kind of the technological breakthroughs that have occurred. CMT is on the cusp, I feel. You know, there's been all this legwork to get things to this particular point, and it really feels like there's this acceleration. And whether it's folks at Jackson Labs, if it's Shift, if it's DTRX, you know, there's all of these groups that are now focused on CMT. And the reality is that they're not in it to fail. These are groups that really want to do a good job, and they understand that there's an opportunity there as well. And so it kind of is hand in glove in the sense that you want things to move forward and they want to be successful as well. And I think that's one of the things that you can really see is when there's a defined path, you really start to feel this acceleration happen. And that's where I believe that CMT is. And it felt the same in 2008, 2009, 2010 in SMA, where it went from a very kind of polar research and clinicians in different rooms to research clinicians, government agencies, and biotech big pharma all in one room saying, guess what? It's time. And I think what you're doing here, bringing everybody together, kind of expanding the portfolio so you have multiple shots on goal is really the right way to do things. So that's what I'm excited about.
Thank you. Thank you. I really feel that momentum. I feel that action that's happening, and it's such a thrill to have you two as partners to help us get it done. Thank you so much for helping to guide the science and guide the foundation in the right direction. I really appreciate it. So this kind of concludes this little Oprah segment of the evening tonight. You all at the beginning met my scientific partner, Dr. Keith Fargo, and now I have the immense pleasure to introduce you to my business partner, Eric Fasnacht. Eric.
Thank you, Susan. Yeah, I'm really struck by this whole presentation. There's so much kind of dense information here, but it just demonstrates where we are. I think to myself, what if this presentation didn't exist? Or what if we weren't having this night? What if this organization didn't exist at all? That whole excitement that Chris just talked about, it just wouldn't be there. DTX wouldn't be working in CMT. Shift wouldn't have the momentum that it has right now. It would just be a big blank slate, and that would be awful. Look at where we are. I've heard the word on the cusp a couple times, and that's amazing. I'm here really to just make one brief announcement. It is a very special announcement, and it's around funding the work that we do. So funding is critical. It's what makes all of this possible. And so we have an announcement that there's a match going on. So the match means that any dollars that are donated to the foundation will be matched up to $400,000. There are two very, very generous families that have put this match together. And so what we want to do is, you know, $10,000 will turn into 20, $100,000 turns into 200. That's where we are right now in terms of our best shot at leveraging dollars to fund the work that we just saw. We're laser focused on this 400 grand right now because we know it'll turn into 800. And so again, Susan, myself, Keith, anyone on the team is available to chat further, ideas around raising those funds. That's going to be critical to our future. Turn it back over to Pat.
But I will tell you, the greatest thing that I could have as a legacy, and the greatest thing I think most of us on this Zoom call could have as a legacy, is seeing a therapy get over the goal line. From my standpoint, if I can pass that down and stop another generation from this devastating disease that affects probably 3 to 5 million people in this world, I've done what my parents taught me, is try to make the next generation better off than mine. So we've got the ability, we've got the power. From the bottom of my heart, I'm very grateful for everyone. I also want to shout out on two heroes on this line, Chris and Charlotte. You are heroes to the CMT community. I also believe that the pharma companies and the biotechs that are jumping on are heroes to this community also. It takes a team. I'm bullish on where we are right now. We can do this and we can win on this. I want to thank you everyone from the bottom of my heart for attending, and I wish you all a good night. Thank you.